Hello, I'm Judith Greer and I'm a Senior Research Fellow at the University of Queensland Centre for Clinical Research, and my main area of interest is in autoimmune reactivity in the brain, particularly in multiple sclerosis. We're doing a range of projects. They're all focused basically on the autoimmune reactivity to one of the major myelin proteins, myelin protolipid protein, or PLP. The type of things we're looking at are why PLP is the target of autoimmune attack in some people but not in others, whether autoimmunity focused against PLP leads to a certain pattern of MS, so lesions in a particular area. What we've found so far is that patients who show increased reactivity to one particular region of PLP, particularly when they carry certain immune response genes called HLA antigens, if there's the correct combination of PLP reactivity and HLA, then we're finding that these patients tend to develop lesions affecting two particular parts of their brain, the brainstem and the cerebellum. And these are important areas in the brain because they control things like balance, but also things like breathing. So we think this is an important area to look at further, and so what we're doing is not only looking at whether we can specifically prevent the lesions from occurring in those places. Almost all of the therapeutic agents that are immunotherapies, so they're targeting the immune cells in MS, they affect pretty much your whole immune system. So they'll kill off not only the cells that are inducing disease, probably in MS, but also the cells that you need to stay healthy. So what we're looking at is trying to develop therapeutic agents that are very specific in their action, so that they'll only target the cells that are actually causing the damage in MS, and not the other cells that you need to, to be able to make an effective response against the influenza virus, or or fight off cancer cells or things like that. The particular method that we're planning on using is one involving making a very slight modification to the naturally occurring myelin protein. You can trick them into making an actual protective immune response if you make the right change. So we're trying to identify which particular parts of the myelin proteins we need to change in order to make these protective peptides, and they're called altered peptide ligands because we're changing one little bit of the peptide. And what we're hoping is that by using these altered peptide ligands, we'll then be able to just target the cells that are causing the damage in MS and leave the rest of the immune system unaffected. We're hoping that the way of modifying these peptides, the technique that we're using to modify these peptides, will be beneficial not only in MS but also in other autoimmune diseases. That we will know which particular APLs will be useful in treating patients in the very near future, actually, within the next year or two. The thing then is to develop it further to get it into human trials, and there's quite a few steps in that process that it has to go through, safety trials and just the testing to ensure that it's not harmful in people before it actually gets to the trials. It may still be 5 or so years before it would get to the stage where it could be used therapeutically.
My name is Shannon Beasley. I am a PhD student with Judith Greer at the University of Queensland Centre for Clinical Research. My project entails the role of patient antibodies in multiple sclerosis, in particular what the antibodies' effects have on the cells that make myelin, called oligodendrocytes. I am culturing oligodendrocytes, which are the cells that make the myelin in your brain and spinal cord, and I'm culturing them with patient antibodies and seeing if the antibodies actually are targeting them directly and killing them. In that way, we'll be able to further understand whether or not the antibodies in MS patients actually do have a pathogenic role in the disease process, or they just happen to be as a bystander part of the disease. The positive outcome from my research will be if we do discover that the patient antibodies are actually pathogenic, we can then target to correct a more effective treatment for patients to alleviate the disease.
My name is Evan Sauer and I'm a researcher at the University of Queensland Centre for Clinical Research. One of the reasons why people get ill in multiple sclerosis is that we think there's an immune response against a portion of myelin called an antigen. So we're looking at ways of specifically dealing with that particular abnormal immune response without affecting the rest of the immune system. And we're doing this with molecules called altered peptide ligands, which will hopefully just target those self-reactive T cells and not affect any of the other ones. I'm looking mostly at the cellular chemistry to see what's going on within the T cells which are recognising this altered peptide ligand, so we can look at different ways to divert them into non-harmful behaviours. So in terms of how far along we've come in this particular avenue of research, the way in which T cells normally respond to their targets is fairly well characterised. We know basically which molecules in the protein in the cell signal to other proteins. So that's all well. The landscape is pretty well mapped out. So now we just need to find out which route is being taken along that map with our particular altered peptide ligands. When this is applied as a therapy, it's really going to be tailored to fit the particular individual based on mostly which T cells they happen to be carrying, and also the way in which the altered peptide ligand is presented to the T cells, and that comes down to genetic background. Solving this problem will give us a good groundwork for addressing other questions. First of all, this sort of therapy would be applicable to many other immune diseases, but also it will tell us a lot more about the underlying cause of MS. But if we get a good handle on this particular part, then we'll know a lot more about what's going on with the immune system in multiple sclerosis,